This diagram shows the signaling process of a receptor tyrosine kinase pathway, specifically through the EGF receptor. RTKs are cell surface receptors that, when activated, trigger signaling in the cell, which cascades, promoting cellular growth and division. When EGF binds to its receptor, the receptor goes through the pairing and autophosphorylation. It activates the intracellular domain. This activation recepts signaling proteins like RAF, a MAP kinase. RAF phosphorylates and activates MEK. Activated ERK phosphorylates cytosolic targets and translocates into the nucleus, where it phosphorylates transcription factors such as CMYC. These transcription factors drive the expression of genes involved in cell growth and division. This pathway is tightly regulated and essential for normal cell function, but can lead to diseases like cancer if dysregulated, as mutations may result in uncontrolled growth and signaling.
DNA synthesis, also known as DNA replication, is a semi-conservative process that occurs during the S phase of the cell cycle, ensuring each daughter cells receive identical copy of DNA. It begins at the origin of replication, where the enzyme helicase unwinds a double helix, breaking hydrogen bonds between base pairs and creating a replication fork. The enzyme primus synthesizes short RNA primers complementary to the DNA template, providing a starting point for the synthesis. DNA polymerase extends the new DNA strand by adding nucleotides to the 5' to 3' direction, using the parental strand as a template. DNA polymerase removes RNA primers and replaces them with DNA nucleotides. The result is two identical DNA molecules, each consisting of one parental and one newly synthesized strand. Mitosis is a process of cell division that ensures equal distribution of genetic material into two daughter cells, occurring after interphase, which includes G1s and G2. Mitosis itself begins with prophase, where chromosomes condense, the nuclear envelope breaks down, and the spindle forms. In metaphase, chromosomes align at the metaphase plate. During anaphase, sister chromatids are pulled apart to opposite sides of the spindle fibers. Telophase then begins, with nuclear envelopes forming around the separated chromosomes and the chromosomes condensing. Finally, cytokinesis divides the cytoplasm, completing the formation of two genetically identical daughter cells. Together, the phases ensure precise replication and distribution of genetic material.